In the name of Allah, the Beneficent, the Merciful. Genetic basis of cancer can be discussed under following headings. Number 1, gain of function mutations in oncogenes. Number 2, loss of function mutations in tumor suppressor genes. Number 3, chromosomal translocations. Number 4, bypassing of apoptosis. And number 5, role of viruses in causing human cancer. Certain proteins serve to regulate cell growth and differentiation. These proteins perform any one of the two main functions to check cell growth. Some proteins cause cell growth, but in a controlled manner so that it does not become explosive and uncontrolled. Still, other proteins facilitate programmed cell death so that cells don't become immortal and live forever. These two kinds of proteins are coded by two main types of genes. Proteins destined to regulate cell growth are coded by protooncogene, whereas proteins that cause apoptosis are coded by tumor suppressor genes. Cancers basically arise from mutations in any of these genes. A gain of function mutation in protooncogenes transforms these into oncogenes, which code for proteins that cause uncontrolled explosive cell growth. The other mechanism is loss of function mutation in tumor suppressor genes. So these genes no longer code for proteins that can cause cell death. Oncogenes, which cause cancer, are actually mutated protooncogenes. For every gene that causes cancer or for every oncogene, there is a corresponding protooncogene that has mutated and fallen astray and is treading the path of uncontrolled growth. So an oncogene is an altered protooncogene. It codes for proteins that speed up cell growth or cell division. For example, a point mutation in a RAS protooncogene that normally encodes a small GTPase. A point mutation in this gene converts it into RAS oncogene with no GTPase activity. This loss of GTPase activity leads to chronic unchecked stimulation of adenylyl cyclase, leading to uncontrolled cellular proliferation. Oncogenes are mutated protooncogenes. Oncogenes and loss of activity of tumor suppressor genes lead to increased cell growth, resulting in cancer. Oncogenes are derived from protooncogenes. Tumor suppressor genes code for proteins that normally suppress cell growth. Cell growth can be influenced by two strategies, either through targeting of signal cascades involved in cell growth or by meddling with the checkpoints of cell cycle, and that's what oncogenes do. So these two mechanisms need to be considered. First, I will focus on signal transduction pathways. To understand this, let's first recap normal signal transduction pathway involved in cell growth. RAS MAP kinase pathway. Ligand binding to catalytic receptor causes phosphorylation of tyrosine residues in tails of receptor which are hanging into the cytoplasm. The phosphorylated tyrosine residues serve as docking sites for adapter proteins like GRB2 and SOS, which consider phosphotyrosine residues as their sofas for sitting. RAS G protein also joins this aggregation of adapter proteins. Here, SOS acts like a money changer and exchanges GTP for GDP on RAS. This serves as energy currency for RAS, which becomes activated, phosphorylates RAF, or mitogen activated protein kinase, abbreviated as MAPKK. This starts a phosphorylation cascade leading to activation of MAPK that translocates through the nucleus and phosphorylates and activates a transcription factor ELK, which activates transcription of genes for cell division. Hydrolysis of GTP to GDP by RAS terminates this cascade. After recap of this normal pathway, now it can be appreciated that mutations at following levels can impair proper functioning of this pathway. A. Growth factor, which is the ligand here, if mutated, can be overexpressed, leading to increased function of this pathway. Secondly, mutation in receptor can cause them to turned on. So that is the ligand, and the second is the receptor that which, if mutated, can remain turned on indefinitely, even in absence of the ligand. This leads to uncontrolled proliferation. Next, the signal transduction proteins may be mutated. For example, RAS gene may code a RAS protein that remains active forever. So that is the RAS protein. It has moved to the receptor. And finally, the transcription factors may be mutated, leading to misexpression in cell cycle. So now coming to oncogenes and cycle. Besides signal transduction pathways, oncogenes may dysregulate cell cycle. Cyclins and cyclin-dependent kinases are proteins that control progression from one phase of the cycle to the next. Different cyclins made constantly during cell cycle during different times of cell cycle control transitions between G1 and S phase, S and G2 phase, G2 and M phase. The activity of cyclin or CDK complex is further regulated through phosphorylation and through cyclin-dependent kinases inhibitors, CKIs. CKIs slow cell cycle progression by binding to CDK cyclin complexes. CDKs are controlled by activating or inhibiting CDKs. Progression through cell cycle is opposed by CKIs. Transition from G1 to S1 is a very important control point in cell cycle. To stop transition from G1 to S1, RB protein binds protein E2F. When it is required that cell be allowed to move from G1 to S1 phase, CDK4, CDK6 and cyclin D appear. Cause phosphorylation of RB protein. As a result of phosphorylation, RB protein dissociates from E2F and cell cycle can continue. To stop action of CDK4, CDK6 and cyclin D, cyclin-dependent protein kinases or CDKIs come into play. These are inhibitory proteins that terminate action of CDK4, 6 and cyclin D by phosphorylation. Loss of function of CDKIs can lead to cancer as they act as tumor suppressors. So that is the control point, and the retinoblastoma protein binds this transcription factor E2F and it stops transition from G1 to S1, and when it is required that the cell cycle should proceed, what happens is that it is inhibited. And how it is inhibited? It is through phosphorylation, which is caused by cyclin D, CDK4 and CDK6. So it dissociates from the transcription factor and now the cell cycle can continue. And when it is required that now the cell cycle should stop, CDKI comes into play, phosphorylates these three proteins, and this leads to cessation of the cell cycle. So there is an MCQ about a newborn with retinoblastoma gene. The child has hereditary retinoblastoma, which is due to inherited mutation in retinoblastoma gene, because RB gene is a tumor suppressor gene.